Okay, picture of it, let's go home.
All right.
Okay.
Okay.
Wait, okay? I still missed what she just said.
Don't you have to always pay attention, it just slips right on by you. So today we're doing metabolism, which has 3 phases: glycolysis, citric acid cycle, and the old name is the Krebs cycle, and electron transport. All right? That'll probably take us most of the time. And what we don't finish today, we'll do on Tuesday, which is up at the top, where it says proteins and carbohydrates and fats. That's to look at the glycolysis, obviously, is looking at carbohydrates. But various proteins and fats can come into this energy activity, series of reactions and capturing the energy and storing it as ATP at various locations. So I talked about the 3 carbons of the glycerol, all right? That comes in, you can see glycerol right there, that comes into a stage of glycolysis. The fatty acids can be broken down via beta oxidation into 2 carbon molecules, which is what our acetyl-CoA is, the acetyl group of the acetyl-CoA. And that can come into that group. So this is a summary of all of those particular components. So if you're getting lost about where are you in all of this, you can come back to this particular slide that kind of is, will help to anchor you. Okay? Now, how many of you have looked at metabolism, at the glycolysis or citric acid cycle at some point? Okay, almost all of you. All right. So what I'm saying here should be a review. I don't think I will go into it in a more complicated component. If you look at your lecture notes, under each component, under glycolysis and intermediate and citric acid cycle and electron transport, is a box with a summary. And that's what you need to know about each of those events. Okay? So let's start in going through with this. So these are the 3 major components again. And remember the whole purpose of this. This is where we end up using the oxygen that we talked about with respiration, and producing the carbon dioxide that is the waste product for that. And we kind of, our very first case study in Bio 430, those of you that had to deal with the mitochondria and the enzymes that are there. So although glycolysis is not our most energy rich, it is probably carbohydrates are typically the highest source of fuel, the most common source of fuel in the body. And glucose is one of our more common simple sugars. When we looked at digestion yesterday, it was the simple sugar that was found in every disaccharide. So we had glucose and fructose, glucose and galactose, or 2 glucose molecules. So the other simple sugars can come into this cycle. In fact, glucose becomes reformatted as fructose before we actually complete it. And the electron transport, I think what surprised me the first time I looked at this was it's the energy of the electrons. I always had the idea is when you release the carbon-carbon bonds, that amount of energy was captured. But it's the energy in the electrons that is transported and finally captured as ATP. It becomes a very physical, there's actually a spinning turbine that's involved in capturing that energy as ATP. So the first sequence that we look at here is what we covered in muscle physiology as anaerobic. All right, this step can occur without the use of oxygen, which is going to happen down here. Without sufficient oxygen available, this byproduct or product builds up and is converted back to lactic acid until there's sufficient oxygen to go back through the process and move it down through oxidative phosphorylation. So this step is considered to be the anaerobic step. It takes place in the cytoplasm, and it's limited as to the amount of ATP that can be produced. All right, so while you can do anaerobic activity with it, it's not going to be as efficient. Let's take a look at that place first. If we're looking at these sequences, and each of these steps is regulated by an enzyme, and where would those enzymes be, how would those enzymes be synthesized? So all of our cells are going to, that make ATP, obviously, are going to need to synthesize these enzymes. So glycolysis is taking place in the cytoplasm. Where is the synthesis of these enzymes taking place?
Right beyond.
No. No. Enzymes are, so that the transcription is going to take place in the nucleus, and the messenger RNA is going to move through the nuclear pores and start translating with transfer RNA coming in. But because these are cytoplasmic enzymes, those ribosomal messenger RNA complexes do not associate with the ribosomal ER, because remember, those are enzymes or proteins that are going to be packaged and always be within a membrane in the mitochondria or in the lysosome or sent out of the cell. This actually takes place within the cytoplasm, not within a membrane-bound vesicle. So each of the enzymes involved in these steps is going to be synthesized via 3 ribosomes, okay, via polyribosomes. Here it is, almost the last day of class, and we're still going back and remembering reviewing stuff from Bio 430. Okay? Because those are, we always think of protein synthesis as dealing with RAR, but we do have enzymes that function within the cytoplasm itself. All right. So, in our very first step, our first couple of steps, we have to use activation energy. Now, I don't know how, we haven't really done chemistry or physics in here, but usually the common image of activation energy is if we've got a potential energy, let's say we're pushing a big rock and it's going to come down here and squash a car, all right? So that's the potential energy, the force that it's going to follow. But before it goes over that cliff, there's a little rim of rocks that we have to push it over. So even to get it rolling, it takes some addition of energy before we can utilize the energy in the mass of the rock falling with gravity. So to start this glycolysis process, that's what this first couple of molecules of ATP is required for. Now our net gain of energy in the process of glycolysis is 2. So although we do end up releasing sufficient energy to store phosphate molecules and make 4 ATP, we have to replace the 2 molecules of ATP that were added to the system to get the whole thing going. All right? So that's where these 2 phosphates come from. We reorganize the glucose. We still have 6 carbons. So that's what I want. One of the first things that I want you to recognize is that we start off with a 6-carbon molecule. I don't know how much you remember about the glucose is a 6-sided sugar, but only 5 of the 6 sides have carbon as their anchoring, and the other corner is the oxygen. So when we reorganize it to fructose, we just reorganize it, but we still have the 6 carbons. Okay, the carbons are numbered, and so that's where you see the 1,6. One phosphate is on the first carbon, and 1 carbon phosphate is on the 6th carbon. All right. So we start out with 6 carbons. We add a phosphate from our ATP. And we add another phosphate. So now we have a mirror image, right, of this molecule. An enzyme is utilized to split carbon-carbon bonds. We're not releasing energy that we can capture. All right, now we have 2 3-carbon molecules, each of which has a phosphate. This is where glycerol, as a component of triglyceride, can come into the pathway, and we can utilize fat to capture energy. All right, is in that 3-carbon molecule. So we have 2 of these, and now everything is going to be done twice. Hydrogens are removed, and don't forget, along with those hydrogens, we have electrons. Okay? Those electrons are what become our energy-rich source, and so we are going to utilize those hydrogens and more specifically the electrons and electron transport. So we have to set those aside because we're not going to use them now. But in removing the hydrogens, sometimes written just more lazily NaDH2, we have now changed our chemical molecule, right? It's no longer the glyceraldehyde 3-phosphate. The 3 is just indicating which carbon the phosphate group is on. With the loss of hydrogen, we still have 3 carbons. We haven't lost a carbon. Now we have 2 molecules of pyruvate or pyruvic acid. We still have all of our carbons and our oxygens, okay? And now, if oxygen is present, we go to the intermediate step. In removing these hydrogens, enough energy was released to make those 4 molecules of ATP. We had to pay the energy back. So our net gain, as far as ATP is concerned, is 2 molecules of ATP. Right now, I'm not going to have you recognize how many numbers of ATP throughout this. The reason I'm emphasizing this here is to emphasize that this is inefficient. It provides energy, but it's an inefficient mechanism if we have to stop with anaerobic metabolism. Okay. This is just showing that if oxygen continues to be absent, the pyruvic acid is converted to, the pyruvic acid is converted to lactic acid, and that becomes our oxygen debt source, the amount of oxygen that we need to go back and continue the pathway. So we started out with 6 carbons when we began glycolysis, and we continue with 6 carbons. Okay? This is just a little bit more of the chemistry of it. A couple of slides that I added last night when I was stuck waiting for my daughter. So you can see how when we go from glucose to fructose, we change the arrangement where the ring formation, but it's still the same molecule. Not the same molecule, it's still the same number of carbons. All right, so we started out with 6 carbons, and we end up with 6 carbons. So down here at the bottom, when we're done with our removal of hydrogens, not showing on the bottom of the screen, but we still have 6 carbons. 2 molecules, each with 3 carbons each. So we're not producing carbon dioxide in glycolysis. So again, if you look at the end of the glycolysis component, and I've given away all my lecture notes so I don't have a copy, there should be a box, or at least a summary if it's not in a box, that says each of the things that I want you to know for glycolysis. Okay? So I want you to know where ATP is produced, where carbons are lost, where we capture hydrogens in our NAD components. So now we're ready to move into the mitochondria, right? And we go through our intermediate step, which is the first time that we actually lose carbon. And we actually remove, by losing, I mean just removing carbon from our molecule. So we have our 3-carbon pyruvic acid. A carbon is released along with oxygen, and we end up with a 2-carbon group that is transported by a coenzyme called acetyl-CoA. Well, the coenzyme is coenzyme A. Okay. And that transports it into the mitochondria. So this is the first time that we create carbon dioxide. And we actually release, how many molecules of carbon dioxide? 2. 1 from each pyruvic acid. Okay. So with glycolysis, we transport hydrogen hydrogen. We are NAD coenzyme. We store energy. As ATP, but no carbon dioxide formation. With our intermediate step, we release CO2. So that's going to account for 2 of our carbon dioxide components. So that's the step at the top. You can also see that additional hydrogen is captured with our coenzyme NAD and transported. So now we enter into the mitochondria. So our coenzyme A helps transport this 2-carbon molecule, the acetyl group, into the mitochondria. And within the inner matrix, remember in mitochondria we have the cristae, and with the outer membrane we have an outer matrix, and then within the cristae we have an inner matrix. And so in this pool are a whole bunch of enzymes now that are going to be responsible for the reactions of the citric acid cycle. Those enzymes, all right, can either be produced by the DNA structures within the mitochondria itself or via ribosomal ER transported to the RER and the Golgi apparatus to the mitochondria. The cycle doesn't mean that these enzymes are organized like a fairy ring in a ring-like structure, obviously. The reason we diagram it that way, but the reason we have the term cycle is because one of our beginning molecules, our substrates for the enzyme, becomes our product. And so we just keep producing the product that keeps this going. So when we introduce 2 carbons, those 2 carbons, we do lose 2 carbons in the cycle. Obviously we have to end up with a 4-carbon molecule. Those 2 carbons that are lost in that are produced as CO2 in that first run through the cycle are not exactly the same 2 carbons that came in. So it takes more than one cycle if we were to tag them radioactively. We wouldn't see them as in the carbon dioxide molecules that went out, but mathematically, 2 carbons are added to a 4-carbon molecule. We form a 6-carbon citric acid, and as we go through the cycle, hydrogens are removed, captured with our coenzyme. Enough, it's either ATP or GTP, but energy is stored, and additional carbon dioxide is released. 2 carbon dioxides for each cycle. However, we're bringing 2 acetyl groups in. Those 2 cycles don't have to wait in turn. There's plenty of enzymes. So they may or may not be happening at the same time, but it's short sequence of each other. And so by passing through the citric acid cycle, we complete the release of all the remaining 4 carbon dioxide molecules. So by the time we're done with the citric acid cycle sequences, all we're left with are the hydrogens to complete our energy transfer. All right? So we will have released 2 carbon dioxides in the intermediate step in the formation of the acetyl group, and we will have released 4 carbon dioxides by the time we complete the citric acid cycle. So I have this diagram already up there on your PowerPoint thing that puts this together. Again, specific enzymes for each step. All right? One step will not occur without the enzyme that regulates the previous step. So I have another couple of slides I put together this morning based on YouTube that I was watching that put it in sequence. But this ties in the pyruvic acid, 2 carbons, The 2 carbons separate from the coenzyme A, which returns to pick up additional 2 carbons. And here is our 4-carbon oxaloacetic acid. Enzyme helps to synthesize those together, and we have a 6-carbon molecule. We reorganize that 6-carbon molecule, so isocitric acid is a supplementary step. And then again, we remove hydrogens and carbon dioxide. Hydrogens and carbon dioxide. Here we can make enough energy is released to make it's equivalent of ATP. It's really GTP, but the equivalent of ATP. Hydrogens. This is another coenzyme. It's not, the hydrogens here are not as high in energy level as the ones that are bound to NAD, and hydrogens again, right? So this has to be repeated, or it's ongoing at the same time. So here we have the acetyl group with 2 carbons, oxaloacetate or oxaloacetic acid with 2, and we form citric acid. Hydrogens are removed with their electrons and carbon dioxide to form alpha-ketoglutarate or glutaric acid. Additional, it's supposed to be an electron receiver down there. Electrons are transported. We repeat that process in making the equivalent of ATP. And so this just continues to go around and around. Now, we're not really going around, it will along in sequence. So what do I want you to know from this component? Again, there's a summary at the bottom of your page. I want you to know the acetyl-CoA. I want you to know that the acetyl group is 2 carbons. I want you to know that it joins with oxaloacetic acid or oxaloacetate, which is 4 carbons. I want you to know that citric acid is 6 carbons. Okay? And how we form citric acid. This should have the NAD over there when I copied it, I forgot to put it back. And then as we go through this cycle, you don't have to know the alpha-glutaric acid or the xenyl or fumaric acid. There's a couple of others that are left in here. But I want you to know that in that process, additional hydrogens are captured, if you will, by the coenzymes NAD and FAD for use in our electron transport. That we lose our last remaining carbon dioxides twice. So each acetyl group, as it passes through the cycle, is going to result in the loss of 2 carbons. So by the time the citric acid cycle is completed for both acetyl groups, we now have produced all the carbon dioxide from our glucose. 6 molecules of carbon dioxide. And when we finish, we're back to our product, which is our starting process again. Now, although this process does produce ATP, if you want all of the molecular structures here, they are. So, it's kind of just a bigger summary. While we do get ATP, the equivalent from GTP, primarily we're capturing a lot of hydrogens. Okay? That's the primary use of the citric acid cycle, are the hydrogens that we capture because that's the energy source. Electrons that are in that, in those hydrogen molecules. Now we're ready for the 3rd step. So we've done glycolysis, which gave us actual ATP, but sent electrons via the NADH. Our intermediate step sent additional electrons. Our Krebs cycle made ATP, but sent additional electrons via the hydrogen and on the NAD and the FAD. And now we're ready for the 3rd step. Okay? And this is where most of our ATP is produced. Now, I don't know how many numbers do you talk about in microbiology.
not many numbers. In microbiome, she just gave the end product. It was chemically toxicity.
Was it 36 or 38, I guess is the number, right?
It was 38, and in microbiome 32 in chem.
Okay, and what I was reading yesterday, they were saying it was like, okay, in prokaryotes it's really 30 by the time you subtract what you lose in this, and in eukaryotes it's really 32, and I'm like, okay, 38 versus 36, and then we're down to 32 versus 30, subtracted from 34 versus 32. So I'm not going to ask again those numbers either, but I want you to look at the net difference, okay? Glycolysis versus citric acid cycle versus electron transport. So if we don't get to electron transport, we're being very inefficient without the use of oxygen. Now, where does electron transport take place?
In the outer mitochondrial membrane?
Along the cristae. Our enzymes for the citric acid cycle are dissolved in the cytosol, or the fluid matrix within the mitochondria. The enzymes for the citric acid cycle are embedded in the cristae. Now, back in the beginning of IL-430, when I first started talking about the cell, when we talked about what would happen if I assigned each of you a number, and all the odd numbers had to go find their next successive even number, and I let you, I distributed it all over campus, and you were blindfolded, and you were trying to bump into, ones were trying to find 2s, and 3s were trying to find 4s, and you just had to randomly bump into somebody, okay? And then I said, all right, now we're going to limit you to the classroom. You're still blindfolded, you're still randomly bumping into your partner. Now, when we talk about the mitochondria, let's take all of the odd number, the 1 and the 3s and the 5s, and you have to be, you can move along the tables, but you have to be in a chair with rollers along the tables. The even numbered people can roam around. So now we've made it even more likely, more efficient, that the randomness is not going to take as long to find the partner. So by limiting the enzymes of electron transport and oxidative phosphorylation to the cristae, then this becomes more efficient as far as time takes for enzyme and substrates localizing each other. This diagram is primarily to show the energy levels. All right, so remember, our purpose is to capture the energy of the electrons. They're coming in at their highest level associated with the hydrogen proton. And then they are released and recaptured by another molecule that has a higher affinity for that hydrogen or the electron, but does not require as much energy to hold on to it. So it's more likely to leave this molecule and go to this molecule. And yet, at certain locations, there's sufficient energy released to pump our hydrogen ions from the inner matrix to the outer matrix. And as it moves from successive energy level, notice how each of these is lower and lower and lower. Finally, we capture it as water. That difference in the energy release is what we store eventually as the energy in the bond between the second and the third phosphate group. So I illustrate this by telling a story. Sorry, women, it's not very flattering to our gender, but since I'm female, I can tell it. Okay? So the hydrogen, or down here we're dealing with the electron, is the girl. The various molecules accepting it are her boyfriends. Okay? She starts out kind of high maintenance. She goes off to a bar, and she's sitting there. She's dressed up for the evening, and this guy comes in that really catches her eye. And as they start to talk, there's a spark between them. She's really attracted to him. And so when he invites her home, she's fine with that. I mean, she's noticed that he's got, some gold chains, and when they go outside and get in his Bentley or whatever. She's impressed, but she wouldn't have gone with him if she didn't like him. And they end up at his mansion, and the next morning, breakfast is brought to her in bed in crystal goblets, and there's 3 10-carat diamonds at the bottom of each goblet. And he says, well, I've got to go to work, but outside there's a helicopter. It'll take you anywhere around the world. You want to go shopping. Here's, my credit cards, but just say my name and the stores will take care of you. Well, this is